the room, and I'm just waiting for people to join. Also, just letting you guys know that the talk is going to be recorded. So if you guys have questions when we have the Q&A time later on, when you come on stage, just know that you're being recorded. Also, one other thing, I'll announce this, or Laura will announce this during the call as well. Just a note that nobody here that we know of is a medical doctor. We are PhDs, and this is not a medical advice channel. If you have questions, if you can not ask medical questions, that would be awesome, in the sense that what you should do or what you should give your pet for medicine as far as disease is concerned, that would be good. So yes, no medical advice will be given on during this talk. Matt, I'm assuming that you concur with that?
Absolutely, yes. I am not a medical doctor, nor am I a veterinarian, so I will not give veterinary advice or medical advice. Thank you.
Otherwise, besides that, this is going to be fantastically entertaining. We are just super excited to have Matt Kaeberlein with us, and we'll do formal introductions just shortly. And thank you, everyone, for being here.
Hello, everyone. I'm just pinging people into the room. Avi, Matt, very excited to kick off our Let's Talk Pet Longevity Summer Series. And this is a special project that myself and Avi Roy, my co-host, are doing for the summer. And I'm very excited to have Dr. Matt Kaeberlein, the co-director. He's a well-known researcher, professor, and also the co-director of the Dog Aging Project. So as Avi said, as a heads up, we are recording. So if you come up for questions, you know that we're using your voice and your photo or image. So you're okay with it, you can come up and ask your question. We're going to do about 30 to 40 minutes speaking with Matt directly. And after that, you're totally welcome to ask whatever questions you have. As I said, this is a summer series we're going to do for the month of August. We have a couple of other very exciting, everybody's a very exciting guest anyways. But I guess I'm just going to get started the call. And I think a lot of me, but if you don't, my name is Laura Minquini, and I am very passionate about longevity. I am building a direct-to-consumer platform for people to try longevity interventions. And I spend a lot of time talking to the scientists, founders, and innovators in the field, trying to also bring them to a general audience so people understand the potential of longevity. And today with me is my good friend and the longevity godfather of my dog Mobile, Avi Roy, who I'm going to let him introduce himself.
Thank you, Laura. My name, everyone, here is Avi, and I'm a biomedical scientist and entrepreneur based here. And I advise funds and startup in health and longevity space in North America, in EU, and in India. And I also work with governments to develop and refine policies that will enable a longevity infrastructure. So without further ado, Laura, would you please do the honors of introducing Matt?
Yes, of course. I am just making sure that I'm getting to our little amazing doc that we did, and here we go. So in today's episode of Let's Talk Longevity, as we have Professor Matt Kaeberlein. He is the professor of laboratory medicine and pathology at the University of Washington School of Medicine. His research focuses on the basic mechanisms of aging in order to facilitate translational interventions that promote healthspan and improve quality of life. And we are talking to him today because he's also the co-director of the Dog Aging Project. Matt, I'm going to let you give a quick intro to yourself before we proceed to some questions. And thank you again for being here with us today.
Sure. So thank you, Laura and Avi, for the invitation. This is actually my first Clubhouse event. So also thank you for pulling me into the 21st century here. So it's great to be here. I've been interested in the biology of aging actually since I was a graduate student. I did my graduate work with Lenny Garenti at MIT, at that time working in single-celled budding yeast. And as my career has progressed, I've gotten more and more interested in which aspects of the biological aging are evolutionarily conserved, and then, how can we actually use this information to have an impact on health and longevity in the real world. And certainly I have a strong interest in translation of this science to improve healthy aging in people. But it occurred to me 7 or 8 years ago that companion animals, and pet dogs in particular, are a really fantastic opportunity to actually bring aging research out of the laboratory and into the real world, hopefully in order to help our pet live longer, live healthier, and thereby enhance the quality of life, not only for our pets, but for the humans who love these dogs as well. And so that's really the goal of the Dog Aging Project, is to understand the biology of aging in pet dogs and to use that information to maximize healthy longevity in pet dogs.
Thank you, Matt. That was actually going to be my first question, is your answer, which was that, can you tell us a little bit about your background and how you got interested in this field? But you already covered it. So I'm going to move on quickly to the second question. By the way, everyone on this call, we have a few questions. Laura and I have a few questions. We have actually a lot of questions, but we're limited to a few questions. And then we'll open the floor up for any questions that you guys have in about 30 minutes or so. So the second question is actually written by my graduate student. It's written in first person, so it sounds like I'm actually asking it, but this is the student that is actually asking it, but it concurs my feelings as well. So one of the things that Laura and I and my research students do is ask researchers a question, which is, how do you define aging? And this may be a trite question to ask, but the variability of answers is quite big from different researchers who are working in this field. So firstly, how do you define aging? And secondly, do you believe that the fact that there is such variability in the answering of this question that is indicative of maybe a problem, or at least some kind of concerns in this field, the field of longevity?
Yeah. So first of all, let me just say that is a fantastic and really important question. So kudos to your student. And I agree completely. I think when you ask that question, you will get a different answer from almost anybody in the field. And honestly, I think a lot of the disagreements that I see happen between colleagues, and even disagreements that I've gotten in with other people in the field, can often, when you finally get to the core of it, come down to, we're starting with different definitions of aging, and what we mean by that. So I think this is a really important question. So first of all, let me say that I think, like most people, have multiple definitions of aging, and it really depends on the context. So I think I naturally, given that I've been working on the biology of aging for more than 20 years now, when I think about aging, my initial sort of process is I immediately gravitate towards biological aging. So that's really the first sort of decision point in this definition, right? Is aging can mean the passage of chronological time. It can mean geriatrics, right? Changes that occur in the elderly. I naturally gravitate towards mechanisms of biological aging, and that's usually what I mean when I use the word aging. And then within that realm of biological aging, I think, again, you can have multiple definitions. And I naturally gravitate towards functional and molecular definitions of aging. And one way to think about that, and this is imperfect, but I think it's the best we have right now, are the hallmarks or pillars of aging, right? These highly shared molecular processes and pathways that seem to affect the biological aging process. That's what I mean when I talk about aging. And then over the last few years, I have very much come to believe that the manifestation of those molecular processes in terms of functional declines that go along with aging are what are really important when we're thinking about translational approaches or translational geroscience. What we really want to do is improve those functional changes that impact quality of life. And so that can be disease-based, like dementia or diabetes or heart disease, but it can also be more subtle things like arthritis or chronic pain in your joints or shoulder. Like I had something called frozen shoulder, which is inflammation of the shoulder capsule. That's not a disease we usually think of with aging, but it really affected my quality of life until I got it taken care of. So I think those are the kinds of things that I've started to value more when I think about aging, is how do these hallmarks or pillars of aging impact quality of life and functional outcomes as we get older.
Thank you, Matt. Yeah, your functional and molecular obviously sync up with anybody who is like myself, a molecular biologist or a cell biologist. So I completely appreciate that. Can you tell us a little bit more about the dog aging project, how it was started and where it is now? A little bit of an overview and coming to where we are today.
Sure. So I'll start with the history, and actually the Dog Aging Project, preceded me. So it was really, I think, the brainchild of Daniel Promislo, who's at the University of Washington with me, and Kate Creevy, who's our chief veterinary officer, and she's at Texas A&M University. And actually they were both at the University of Georgia at the time, and basically got interested in the idea that dogs could be a very powerful genetic model and environmental model to understand what are the factors that influence aging. And so they started a network. It was an NIH mechanism called an R24, which is a network grant to bring together people to think about a topic and come up with next steps. And so they had the Canine Longevity Consortium, and that's where I got involved and had sort of initial discussions with Daniel and Katie and several others. And then when we recruited Daniel to come to the University of Washington, that's when really we started to really, I think, put the foundation in place for what is now the Dog Aging Project. And for me, so Daniel and Katie were really, I think, interested in, like I said, genetic and environmental correlates of aging. For me, what really got me excited about the Dog Aging Project was when I made the connection in my head that not only are dogs very powerful for understanding correlates with healthy aging, but there's actually a real opportunity to slow aging or even partially reverse aging in our pets. And I've always been a dog person. I've had dogs all my life. And I think, like many people, I love my dogs and have considered them part of my family. And it was just this moment that clicked for me that if we could actually have an impact on the healthy longevity of family members. And I think the statistics are that more than half of pet owners consider their pet part of their family. That can have a huge impact on the field and on the quality of life for people. And so that's what really got me excited. And we can talk more about why dogs are a particularly powerful system in which to test interventions for aging, but that's what really got me excited about it. And so then Daniel, Katie, and I really got together and decided if we're going to go for it, we need to go for it. And we started outlining what the Dog Aging Project was going to be, trying to seek funding. It took really probably 4 or 5 years to get the first big NIH grant to build the Dog Aging Project. And that happened, I guess, about 2.5 years ago now. And so now what we've got is a platform that really has two major goals. One is a longitudinal study of aging, which is just observational. And the goals are really to understand what are the most important genetic and environmental components of healthy aging in pet dogs. And then we also have an intervention component, which our first clinical trial is called Triad for test of rapamycin in aging dogs. And so I think these are mutually beneficial goals, right? One is to understand aging, that's the longitudinal study. The other is to do something about it, and that's the clinical trials. And that's where I see us going into the future is continuing to, we've created the platform now, and I think now we can continue to move on into the future. We're going to get a ton of really cool data over the next couple of years. But hopefully we won't stop with rapamycin. Hopefully we'll be able, or other people will be able to use this infrastructure to test other interventions to get to the point where we can actually have an impact on healthy aging in pet dogs.
Thanks for that, Matt. I had my set of questions, but I think if Avi agrees that maybe it's important that you go straight into saying why the dogs is such a good model to prove some of to study and then to prove some of these trials on before moving to humans. Because can you explain this? I think for some people it is obvious, but it is important that people know.
Sure. Yeah. So there are actually lots of reasons, but I think some of the big ones are, first of all, from a public recognition of the field perspective. I think that this has a huge potential, right? I think all of us in the field recognize that among the general public, most people don't have an understanding of where the biology of aging is today. I think, honestly, most of the general public still thinks of it as science fiction or snake oil. And there are a lot of reasons for that, but I think that's just a fact. And I think that it's if we're successful at understanding aging and ultimately having a clinically demonstrated intervention that increases lifespan and health span in pet dogs, I think that'll really be a watershed moment for the field. And it will really shift the way that the general public views aging biology. I think it's going to be much easier for people to understand that aging is actually malleable once we can do it in their pets. So I think that's not a biological reason for why dogs are a great model and opportunity, but I think it's a really important one for the future of the field. I think when you look at the biology, there are also some obvious things that are important. One is dogs age faster than people do, right? We're all familiar with this idea that one human year is about 7 dog years, and that's not perfect. If you're interested, we can talk about what a little bit more about that, but it's pretty close. It's close enough, and really that just means dogs age about 7 times faster than people do. So the importance of that is that you can actually do experiments in a reasonable amount of time. A longitudinal study that would take 3 decades in people will take you 3 or 4 years in dogs. And that's much more amenable to both the pace of scientific research and the sort of typical grant timelines. Most NIH grants are 5-year grants. So you can actually accomplish stuff in a reasonable amount of time. For our clinical trial of rapamycin, it's a 3-year clinical trial where we are statistically powered to detect lifespan as the primary endpoint. You just can't do a lifespan study in healthy older people. It's just not feasible. So that's really important. I think the other big one is that dogs share our environment. 99.9% of the preclinical studies in model organisms in this field have been done in the laboratory. And in the laboratory, we intentionally try to control the environment as much as possible to reduce the noise and enhance our opportunity to see significant outcomes. But the consequence is you lose all of the environmental variability that humans experience on a constant basis. Dogs, pet dogs, share the human environment in pretty much every way. The one area where it's not quite a great match is diet. But other than that, they really experience all of the same environmental factors that we experience and that almost certainly have a huge impact on the aging process. I think the current estimates are that about 75% of longevity determination in people is environmental or at least non-genetic. So I think we all recognize that environment's really important, but we don't have a good way to study it in the laboratory. And pet dogs give us the opportunity to do.
I am very glad that you mentioned how it's the one first thing of why showing that longevity science or being able to extend lifespan matters with pets, simply because when you think of it, I think last year or 2 years ago was the first time that Americans spent more money on their pets than they did babies. And millennials are having dogs instead of children. And the other part is one quote that one study in 2018, which shows there is a high rate that Americans are spending in on their pets than their food or medicine for themselves. So we are naturally more propense to do something for our furry loved ones than we are for ourselves, even when it comes to our health. And people will be willing to do anything to extend the life of a dog, simply because it is, as I think we had this little small discussion before, when you get a dog, you do know that you sign up for the joy of having that companionship, but that it's very limited time. All of us would do anything to have them longer with us. So in terms of messaging and perception, it's so crucial, because if people see a dog living longer or that we did extend life, then it's going to be very easy for people to believe, oh, then it can happen to us too. With that said, I wanted to ask you about, go back to you explaining the rate of aging of dogs, something like this 7 years per year. You said that it's not precise, but it's very close. Can you go about say a bit more about that?
Sure. Yeah, absolutely. I think maybe the first thing to say is this goes back to the original question, which is how do you define aging? So I think part of the challenge here is that we don't have a consensus definition of biological aging in the field. And so, given that, we don't have quantitative ways to measure biological age or biological aging rate. And I'm going to take a second just to expand on that a little bit because I think some of the people listening may not be obvious to them the nuance there. So we all understand chronological age, right? That's just the length of time from when you were born to where you are now, right? And your chronological lifespan is just the length of time from when you were born to when you die. The important point here is that biological age doesn't necessarily have to match chronological age. If the biological processes that we are defining as aging, these hallmarks or pillars of aging, they happen at faster or slower rates in different individuals. That's true in every animal that we know of. So the same 40-year-old person or people, two 40-year-olds, can have very different biological age. And we all know this. Like, I think all of us have people in our own lives that biologically are older or younger than their chronological age. And typically that goes along with health. So if you look at dogs, are really interesting because their biological aging rate is strongly determined by body size. So big dogs age faster biologically than small dogs will live longer, and for most diseases of aging, develop those diseases of aging later in life. So this is actually a really good case example where the rate of biological aging is very obvious and very different across small dogs versus big dogs, even though their chronological age can be the same. So this idea that 1 human year equals 7 dog years is, first of all, not perfect across just body sizes in dogs. So that metric is going to be different in a Chihuahua versus a Great Dane. The other thing that's imperfect about that metric is that it's not linear. And what I mean by that is that early in life, dogs seem to age even much more rapidly than 7 to 1. Maybe it's 12 to 1 or something like that. And then as they get older, their rate of aging relative to people becomes less than 7 to 1. And that's still being worked out. So I don't think we have, again, really good quantitative ways to assess that. But it does seem to be the case that 7 to 1 ratio is not going to hold across the entire lifespan and is different early in life versus late in life. So it's just more complicated than the simple idea that 1 human year equals 7 dog years. But having said that, again, it's pretty close. I think on average, that's not a bad rule of thumb, just to get the idea across that dogs age about 7 times faster than people do across their entire lifespan.
Yeah, thank you for that, Matt. The kind of the rule of thumb. I want to actually dig deeper into what you were saying, because you came really close to biomarkers of aging. So going back to the the dog longevity project, you pointed out that over the next few years you're going to have a whole bunch of epidemiological data, and then you can also randomize this stuff and actually look at testing and words that I'm talking about, Cochrane collaboration, but really testing treatments is what you're going to look into. So my question is that how are you measuring aging in your dog cohorts, and so that you can actually ascertain like what environmental effects they're having and things like that? What endpoints are you looking at?
Yeah, really good question. So let me start with the clinical trial, because I think that's easiest, at least to start. So there, our primary endpoint is lifespan, and the study is really designed to ask whether rapamycin can increase lifespan in middle-aged dogs of normal health status, okay? They have to be larger dogs as well, because as we just talked about, big dogs age faster than small dogs. So that's the primary endpoint. But then for secondary endpoints, we're really trying to look as broadly as possible at age-related outcomes. So all the dogs, for example, will get echocardiograms every 6 months to look at cardiac function. Does rapamycin improve cardiac function with aging? We'll be looking at activity, so the dogs will be fitted with activity monitors. We'll be looking at disease incidence, cancer, kidney disease, dementia, basically every age-related disease that you can think of that dogs and people both get. So the idea is really to take a broad functional and disease-based approach to assess, is this intervention actually impacting the aging process functionally. And then we'll also look at molecular markers. Every 6 months, at the time the dogs come in for their exams, we'll collect blood for standard blood chemistry, blood metabolome, epigenome, and then we'll also collect feces to look at the fecal microbiome. So to try to get a little bit more of a systems biology perspective on what's changing with age in dogs, and how does the intervention, rapamycin in this case, impact those molecular markers. We're trying to be relatively agnostic with respect to the current sort of biomarkers that people in the field are most excited about. We're not going in with the assumption that the epigenetic clock is going to be the single best measure of efficacy for interventions. We're really going to try to let the study tell us whether that's the case. And I think this is a bigger challenge in the field right now, just to take a step back and talk a little bit more broadly, that in my view, too many people in the field have basically decided that epigenetic clocks are measuring biological age. And I think that's a problem, because once you start looking under the lamppost, it's really hard to stop looking under the lamppost. And you miss a lot of important biology. And unfortunately, I feel like that's what has happened in the field right now. Eventually, it'll get figured out, but I worry that we're spending a ton of time looking at the epigenetic clocks and not looking at other stuff that might be equally or even more important and potentially diagnostic for aging rates.
That's fantastic. So, like the NSA, you're basically collecting everything and then whether now or in the future you'll figure it out as to what we can use that data for and how much of that is actually useful.
Yeah, although let me chime in for a second, because yes, we're trying to collect as much as we can, given obviously every study has constraints, right? You can't collect everything, but we're collecting as much as we can. But I want to come back to the idea that this is my personal sort of view on this, but I really feel that the functional measures are the important things to look at. So, if we see that rapamycin makes dogs live longer and their hearts are working better and their kidneys are working better and they're not getting dementia, right? And they're more active, but it doesn't change the epigenetic clock. I'm not going to say the intervention didn't work because it didn't change the epigenetic clock. I'm going to say it worked and the clock is broken. And unfortunately, I think too often people take reverse view in the field right now. It's really what's important. I don't care if you change my epigenetic clock. I care if you make me live longer and healthier. And I think we need to get back to that a little bit.
Matt,
Absolutely.
I just wanted to quickly ask, what do you think is equally or more important than the functional measure of the epigenetic clock, in your opinion?
Sorry, I'm not sure.
You're saying you cannot use the epigenetic clock as the one measure that shows success. And as you mentioned right now, if you did manage to reverse something, it is the outcome that matters. But what other things are you looking at that you find is equally as important, or maybe even more?
So that's absolutely a fair question. So I guess I would say that I'm not convinced that there is anything better than the epigenetic clocks at this point as a single sort of type of biomarker. And I didn't mean to give that impression. I think that the epigenetic clocks, there's a lot of data to support the idea that they're going to be valuable. I think where my concern lies is that too many people have assumed that anything that changes the epigenetic clock in a certain direction is actually having the effect on biological aging that they want it to have. So I'm not saying that there's something better right now. I think what we really need to do, though, is continue to develop other types of biomarkers so that we can actually, once we start to see how interventions affect functional outcomes and healthy longevity, we can figure out which are the most predictive. My intuition is that it's going to be a combination of types of molecular markers. So these sort of multi-omic clocks, so to speak, that some people are thinking about, which could include, things like blood metabolite or metabolome changes, a subset of inflammatory biomarkers, and even maybe some morphological changes. So we haven't looked at all at this in dogs, but there's some pretty interesting data in humans that even changes in facial features, which we all recognize as a part of age, that those can even be predictive of health status, not just chronological age, but actually health status as we're getting older. So if I had to guess, my guess is that it's going to be a combination of these markers that will be most predictive and most useful. I think the other sort of nuanced point that it's worth making here is that a biomarker of aging, in my view, is not particularly useful unless it's actionable. And what I mean by that is that, does it help you to know that your epigenetic clock says you're 30 when your chronological age is 7? Is that useful? Maybe it'll make you feel better, but it doesn't really tell you what you should or shouldn't be doing to impact your health outcomes going forward. What we really want are biomarkers that are useful for predicting efficacy of interventions. And ultimately, what we want to get to is a personalized set of biomarkers, right? So that we can actually personalize geroscience interventions, because I think we all should recognize, right, that everybody's genetics and environment is different. And the sort of optimal dose of rapamycin or metformin or whatever your favorite aging intervention is, for me, might be very different for you. And ultimately, what we want to get to is the point where we can use these biomarkers to predict at a personalized level what are the actionable things that you can do to give yourself the best opportunity for long, healthy life.
Thank you, Matt, for that. Everybody in the room, we will be opening it up for audience questions very soon. Laura and I just have a few more questions before we jump into audience questions. So hold on to your hats just for a little bit longer, and just remember that this show is being recorded. Matt, continuing with that theme, and you have a background in. I just had a quick question, and it's a double-barrel question before I hand it over to Laura. And the second part is not necessary, but I'm just kind of tacking it on. It'll make more sense when I ask it. Is there anything unique or unusual about the aging process in dogs when compared to other organisms? And can the findings of the dog aging project, can you extrapolate the results towards human longevity and human aging ultimately? Not that it is a necessary part of this dog aging process. The conclusion of the dog aging process leading to a longer lifespan for dogs is in itself fantastic. But can you actually extrapolate from that? And is the dog aging process in any way, shape unique compared to other organisms?
Sure. So the short and simple answer is yes to both of those, but I'll expand a little bit. I'll start with the second question, which is, will anything we learn in dogs be relevant for humans? I think it's clear that the biological aging process in dogs is quite similar to that of humans. I think even if you just look at the types of diseases that dogs get with age, it matches almost perfectly with the types of diseases that people get with age, the functional declines that go along with age. And even, the little bit that we know about things that impact functional outcomes and lifespan in dogs also impact functional outcomes and lifespan in people. The two obvious ones being exercise and obesity, right? So it's hard for me to say as a scientist, right, with 100% certainty that the biology of aging in dogs is identical. In fact, it's not going to be identical to the biology of aging in people. But I think everything we know gives us confidence that much of the biology of aging in dogs is going to be shared with people. And so I am equally confident that a lot of what we learn from the Dog Aging Project will be relevant to the biology of aging in people. Now, those are going to be hypothesis generating types of knowledge, right? So we may see a relationship between a particular gene or a particular environmental component and health outcomes in dogs that we would then hypothesize will be relevant in people. Obviously, that requires additional research to go and test that hypothesis. So certainly I'm not arguing that just because it works in dogs, it's going to work in people. But I think it certainly gives us a degree of confidence that that's probably the case, and we or others can go on and test those things in people. Now, you asked, are there differences in dog aging versus human aging? And the answer, of course, is yes. And that's true for any animal. There's this older idea from George Martin, who is the grandfather of geroscience in a lot of ways, of public and private mechanisms of aging. And I think it's a really nice concept that when you look across different species, there are aspects of biological aging that are public, that are shared, maybe across all animals. And then there are aspects that are private to each species, depending on their unique genetic and environmental makeup and how evolution has shaped those animals. And yes, there are going to be public and private components to aging between dogs and people, just there are between people and mice and worms. So the key is to figure out which are which, and I think we've got a pretty good idea already within the field of that. Specifically in dogs, I can point to a couple of examples. So one example is that dogs don't get a lot of vascular disease. When you think about cardiovascular disease with aging, whereas obviously that's a major killer in people, and they get a lot more cancer than people do with aging. So there are differences, at least in the disease presentation. Does that mean there are molecular differences, or is it just that, because of the unique features of dogs and people, that the way those molecular aging processes manifest themselves as functional declines and diseases are different? I think we don't know. The other area that I'm really interested in right now is cognitive function and cognitive changes with aging. So just like in people, dogs show cognitive decline with aging. A subset of dogs will go on to get dementia. That dementia looks at least a little bit like Alzheimer's disease. And so that's really interesting. And we've got an active and expanding project to look at dementia in dogs. The thing that I'm most fascinated with is at least the initial indications there are that this body size relationship I talked about before, big dogs live shorter and appear to age faster than small dogs. That seems to be true for lots and lots of different age-related diseases and functional declines. At least the preliminary data suggests that might not be true for body size. In other words, big dogs, and again, this is preliminary, but big dogs may not actually show signs of cognitive decline and dementia any faster than small dogs do. So that might actually suggest, that there's something special about the brain and the relationship between body size and aging. That's a long-winded answer to your question of yes, there are differences, but I don't think those differences are so big that it's going to limit, the information that we get about potential causes of human aging.
Thank you, Matt. I am going to ask now, in specific to the dog aging project, where are you at with it? And can people still enroll their dogs? I know that it is only in the US for now. And I have a question after those, you answer those questions. When you're looking at enrolling the dogs, as assuming you're looking at different breeds, different stages in life, but are you also looking at the environment that the dogs are living in? Are they living with professional owners? What are their owners? If they live in the city or if they live in the countryside, socioeconomic status, also race, things like those. Are they having any bearings on your studies right now?
Sure. Yeah, all good questions. So I'll start with are we still enrolling dogs? The answer is yes. And I absolutely encourage any of you who are interested in participating to go to dogagingproject.org and nominate your dog. It's a super simple process, 3 or 4 questions. And then once you do that, you will be invited to create an owner portal within the website where you will then be asked to complete what we call the Health and Life Experiences Survey. And that is basically to address the question, Laura, that you just asked, to get the data on the dog's health history, its environment, activity levels, diet, basically capture as much information about the dog and its home environment as we possibly can. And that's a huge amount of data that we've already collected, which is, I think of it as the tip of the iceberg because we're just starting to analyze it. But yeah, absolutely. What you asked, the answer is we try to capture as much of that environmental information as possible through this Health and Life Experiences survey. We also have a cognitive survey, which will give a feel of cognitive function in the dogs that the owners will be asked to complete at the same time. At that time, once they complete the Health and Life Experiences survey, they are also asked to send us the electronic veterinary medical records, and that can come from the owners or from the veterinarian. And the pairing of those two things gives us a really, I think, unique insight into current health status, prior health status, and all of these environmental variables that we capture in the survey. And that's the base group in the Dog Aging Project, what we call the PAC, the Dog Aging Project PAC. People who have completed the Health and Life Experiences survey and been invited to send us their electronic medical records. So that brings me now to where are we now? So we have spent the last 2.5 years basically building this machine to do the science of the Dog Aging Project. So we've created the infrastructure, and anybody who has ever been involved in a big science data project will understand, how immense the challenges are just on the data side for building the infrastructure to do what we want to do. And then we've also set up the clinical component of the Dog Aging Project. So the way this works, there are right now 32,000 dogs in the pack, a little bit more than that, actually. So this is already a pretty large longitudinal study of aging. All of those dogs in the pack are part of the longitudinal study of aging. So 32,000 plus dogs. My goal is to get that up over 100,000, and I think that's doable. From the pack, right now, we are funded to select 10,000 dogs for genome sequencing. They become what we call part of the foundation cohort. So those owners will have a kit shipped to them where they swab their dog's mouth to collect cells, and then it comes back to us for genome sequencing. So that's the genetics part of the Dog Aging Project. For 10,000 dogs in the pack, we'll have full genome sequencing. And then 1,000 dogs are selected for what we call the precision cohort. That's the systems biology cohort. So every year, those dogs will go to their veterinarian, they'll take a sample kit that we send them to the veterinarian, and we'll collect, along with the veterinary exam, blood chemistry, blood for serum microbiome, and serum metabolome, and feces for fecal microbiome. And blood epigenome. So we'll get those 3 sort of omics types of data for 1,000 dogs every year in the precision cohort. And obviously, this will be done on an annual basis. So over time, we'll be able to see how are those things changing as these dogs age and match that up with their genetic and environmental data. And then there's the clinical trial. So that's Triad. Those dogs, so for the other cohorts, any dogs, any size, any breed, any age. So it's really open to all dogs. for Triad, the rapamycin clinical trial, dogs have to be at least 7 years old and between 40 and 100 pounds to be eligible for Triad. They also can't have a pre-existing age-related disease. So this is a clinical trial of healthy aging. It's really, in my knowledge, the first and only true clinical trial of healthy aging in dogs. And the goal, as I've already talked about, is to determine whether rapamycin can slow aging in pet dogs. So we're selecting 350 dogs right now. We're funded to randomize. Up to 500 may come in for their initial visit because some of those dogs will be excluded based on pre-existing conditions. So 350 will be randomized, half in the placebo group, half in the rapamycin group. It's a 1-year treatment period, then 2 years of follow-up, so 3-year total clinical trial with lifespan, as I've already talked about, as the primary endpoint. So those dogs will come in every 6 months. So that's the big picture overview of the Dog Aging Project and where we're at now. We've just had the first few thousand dogs from the pack reach their 1-year anniversary. So the owners every year are asked to update the health and life experiences and cognitive surveys on an annual basis. So we've now shifted from being a cross-sectional study, meaning one time point per animal, to a longitudinal study where we're now starting to collect data on the same animals spread out over time every year. And so that's super exciting, and we're just now digging into the data from HLES, and I can just all I can tell you right now is it's super interesting and exciting and fun. And we've already found some unexpected things that I think are going to be really neat. And then we've also invited a few thousand dogs to Foundation for genome sequencing and a few hundred dogs to Precision, and we anticipate fully enrolling those cohorts. Triad, we've had the first dogs now scheduled for their screening appointment, and I think we'll have the first dogs randomized into Triad within the next couple of months. So we're not as far along as we hoped we would be 2.5 years ago. But I think given everything that's been going on in the world for the last 18 months, we're in a pretty good spot. And honestly, I'm just really happy with where we're at and the potential that I see for what's going to happen over the next few years.
Yeah, I watched a video where you mentioned that obviously it was difficult. You can't ask owners to be bringing their dogs to pets when we're in the middle of a pandemic. So
Right.
sure, it happened. It's taken into consideration in anything that's been done. And I can't wait until you go global, or at least to Canada, because I would love for I just messaged Avi saying, I need to start doing all of this for my dog.
Yeah, that's probably one of the top things on my list that I would like to see us do is be able to go truly international. There are some logistical challenges with that, but honestly, right now it's all about money. We're constrained in what we can do just with the grant that we've got. If there's anybody on this call or anybody who knows somebody who would like to donate to the Dog Aging Project to make it an international project, I would love to see that happen. So I'll just throw that out there on the off chance that somebody's listening.
I feel that Nathan, who's in the call, will figure out something for you. He's the expert at that. Everybody know that they can come and ask questions. I am going to ask Matt one last question. So I'm going to start bringing people up. Feel free to come ask your questions. Well, I wanted to ask you, because you have a dog named Dobie, right? What is his breed again?
He's a German Shepherd.
It's very simple for the consumer, and because I know people always ask about their own health. What is your longevity protocol for Dobie?
Yeah, so it's a good question. So again, I'm going to first of all say what I said at the beginning, which is I'm not a veterinarian. So nothing that I say should be construed as veterinary advice. Honestly, it's funny because obviously I get asked this question a lot, as do almost anybody who works in this field and talks to the general public, right? In some ways, it's a little bit disappointing. My answer is going to be a little bit disappointing because it's nothing that you wouldn't expect, right? If Dobie is at appropriate weight for his breed, he might even be a little bit underweight. We take him for walks at least twice a day. We take him to the veterinarian on a regular basis. We give him lots of love. Honestly, I think that's probably one of the most important things. There's a lot of interesting data that interactions between owners and dogs have important physiological effects on both the owners and the dogs. And so I think giving your dog lots of love is a great health span promoting strategy. But I honestly can't tell you that there's any particular supplement or intervention that I would recommend people giving their dogs at this point. The one thing I would say is there's a lot of people out there who have very strong opinions on diet in dogs. And you can find people who will swear up and down that a particular diet, is the best diet for healthy longevity in dogs, whether it's raw meat or whatever. The one thing I would say is there's not much data to actually support those strongly held opinions for any particular diet in dogs. And that's something that I hope that will come out of the Dog Aging Project in the next few years, are some real it'll be correlative at this point, but some real correlations between diet and other environmental components and health outcomes and disease in dogs to actually give some data. I do think it's important, though, to explicitly state that, at least from the longitudinal study, those are going to be correlations, right? And correlation does not equal causation. So while I think we'll be able to say dogs that eat a particular diet, or maybe even a particular brand of dog food, tend to live longer or shorter, be healthier, more or less healthy, it's still not going to be proof that those interventions or those dietary changes have an effect on longevity. That's the difference between the longitudinal study and a clinical trial. If we see that rapamycin treatment hits endpoints, whether it's lifespan or heart function or activity or cognitive function or whatever, then we'll have a pretty good case that there is a causal effect from that intervention and the outcome, because it's a double-blind, randomized, placebo-controlled clinical trial.
Thanks, Matt. I know we all want a pill or something magical, but even for dogs, there isn't such.
Not yet, although I try not to use the word magical, because that has the connotation, right?
Yes,
I understand what you mean, but I try really hard not to do that, because I think sometimes we want to get away from the idea that changing the biology of aging is something other than rigorous hard science, because it is rigorous hard science.
It is.